Good morning. Hello, students. Today I am going to teach through this video lecture, and you know that we are going through this unit 3, and although we are covering some part of this cell cloning and selection, but today I am going to teach you about application of animal cell culture for in vitro testing of drugs, and also testing of cytotoxicity of environmental pollutants in cell cultures. So to do that, we should understand that MTT assay. So the MTT assay is one of the technique through which we can understand the effective drugs or the concentration of effective drug. So for that we need MTT. MTT is a chemical also called as tetrazolium salt, and this is the formula for overall this MTT. The structure of MTT is like this, and after reacting with mitochondrial reductase, it forms again a color complex called pharmagen, and this MTT is a kind of mimic of NADH, and after reaction with the enzyme present in cells, it gives a purple color complex which can be absorbed by a spectrophotometer based ELISA plate reader at 570 nanometer. So if we have a drug called A and we want to test it in cancer cells, and we want to know the concentration of drug A that which is effective, which could be effective on the cells with respect to control. So for that we need 196 well plate, cell culture plate, and MTT solution which is made in 5 mg per ml in 1x PBS. Then also the drug of different concentration, or you can have different drugs at one concentration or some concentration. Then we also have we should also have drug control. If the drug is dissolved in other than water or the solution which is not present in cell culture media, then we need to keep one drug control with that solvent. And then apart from that, we can have we should choose a cell line which if the drug is targeting oral cancer, then we should choose oral cancer cell line. If it is targeting another part of body like ovarian cancer, so we can choose HeLa. In such a way we can choose a different cell line for doing this MTT assay. Apart from that, we should have a MTT pipette set, then aluminum foil, dimethyl sulfoxide, ELISA plate reader. So first we have to see the cell culture running cell culture flask or plate, whether it is confluent. If it is right confluency, we'll do passaging, and through passaging we'll count the we'll count the cell number, and based on this counting we'll go for a certain number of cells to be seeded that I have already explained you in previous class. And then we'll go for seeding, and that's the seeding. After seeding, we'll keep this plate in an incubator at that incubation temperature and condition. Then after that, in next day, or you can say after one another day, we can check the confluency of the cells, and then if we can think that okay, cell is ready for the treatment, so in parallel way we can prepare a drug, or we can dilute the drug in different concentration, the desired concentration whatever we want to test it, and then we'll go for the treatment of the drug in the seeded plate. Then we'll incubate it again for the days or the hours, how many hours we want to test, whether it is 24 hour, 48 hour or 72 hours. And after the incubation time over, we'll take out the plates and then go for adding MTT in that. We'll add MTT in two way, either of the two way. Either we can remove the whole drug added media, or in the same drug added media we'll add 10 microliter of the MTT solution. So and after adding MTT, the plate should be wrapped with aluminum foil, because MTT is a light sensitive chemical, and then we'll keep it in incubator. After 2 to 3 hour incubation at 37 degrees centigrade, we'll go for adding DMSO. And before adding DMSO, again we have to remove this MTT added media and add the DMSO. Immediately after adding DMSO, you can see the change in the color, which will be a purple color complex, as I told you in previous slide. The formagen will form and it will give a purple color solution, and here you can see it is less purple color, means more cell deaths are there, and here you can see high purple color means cells are alive in this area. But the exact data we can get through spectrophotometer reading, means like a laser plate reading in terms of in Excel sheet format, and then we'll use this data for analysis and plot the graph. Then this is an example from a paper which I have taken for explaining this. So in that paper, the author has used this 5 fluorocytosine drug of different concentration in terms of millimolar, for testing its efficiency on A549 cells. And after that they have plotted this in terms of cell viability, and so this way they have found that okay, this concentration is which concentration is working more, and they have used different cell types, like this is a wild type, and this is thank you.